You doing, Dr. Adam?
I'm good. How are you doing?
That's Friday.
It is Friday. We still have a lot to do.
So why?
3-hour car ride.
Oh, no, Jack, to your friends. I
I'm
like
gonna hang out with the poodle for the weekend.
poodles.
The poodle.
Oh,
But he'll only be the poodle for 2 more weeks because then she's getting a puppy and then he will just be a poodle.
we have a
Two.
half poodle. It's got a personality.
Well, poodles are real smart.
Yeah,
They don't shed either.
No,
Really?
they're
That's
allergic.
nice. My dog sheds everywhere. I gotta sweep up so much dog fur.
Okay, welcome everybody to our Friday science seminar. I am Anna Oliveri, the chair of the chemistry department, and I'm going to be introducing my colleague, Dr. Chris Babaco. He got his bachelor's degree here in Oregon at Willamette University, and then got his PhD at UC Davis, where I did my postdoc, so look at that, we overlapped. Then he went off to Columbia College, where he was a faculty member before he came here to SOU, and now is our analytical chemist on staff. And so he will be telling you about his research today.
Thank you, Anna. Welcome, everyone. Thank you so much for joining me. I'm Dr. Chris Babaco, and I'm going to be talking about analytical chemistry. So for those of you who are less familiar with the wide world of chemistry, in the same way we could think about different disciplines in any other field, we can think about differences in chemistry. So we have our lovely organic chemists making us fantastic new molecules, Anna and her rocks and all other inorganic properties. But what I do is analytical chemistry. And broadly, that means I'm interested in being able to tell you what is there and how much of it. And my particular focus is on how we can use the tools that we talk about in analytical chemistry to actually address environmental problems. And so we're going to look at 2 of my past projects today. One has to do with the flooding of a conservation area, and the other has to do with the Almeda fire. So we're going to start with an examination of a study I did before I joined the faculty here, looking at the distribution of cocaine and its metabolites in the Eagle Bluffs Conservation area. So when we're thinking about why one might look at cocaine, it's awkward, but whatever, right? When we're thinking about why we might choose this topic, there's actually an increasing amount of research being done on the prevalence and occurrence of pharmaceuticals and what are called drugs of abuse, right? So illicit compounds, in part because most of them move through our waterways and end up in some kind of a receiving body, whether that's a creek, a lake, an ocean, right? So when we have done studies on these compounds in the past, there was a fantastic review. This is from 2013, so arguably out of date, right? Part of what we notice is that there is a bit of a regional distribution in where we see these studies being done. We see a lot of them in the United States. We see a lot of them in Western Europe. We see a fair number of them in Southeast Asia, principally in China. But when you are looking at the United States, there is a ton of work that is done on the Great Lakes. There is a ton of work that is done in California, and there's a lot of work that's done in Colorado. So where I was in Missouri is one of the white spots you can't see on that picture. Okay? Part of the interest in understanding these compounds comes from what happens when we go looking for them. One of the tricks we sometimes think about is if you don't go looking for it, you don't know if it's there, right? So there was a study that was done in the Catalonia region of Spain, right? So we're looking around Barcelona, and what they did is they studied an entire river system. And they did 2 things. First, they looked at wastewater treatment plants, and then they took grab samples. They went out, collected water, and tested it to see if there was cocaine there. And that's all those green triangles. And what I'm going to do is I'm going to highlight the places in which they found cocaine. They found it at their first sampling site, and then their next sampling site, and their next sampling site, down and down until everywhere they looked, they detected these drugs of abuse, right? They're incredibly common pollutant in our environment. Okay? Where I was in Missouri, and I'm going to guess not all of us are as familiar with Missouri as I had become, right? This is the state of Missouri. Columbia is right in the center, and that's where Columbia College was located. And I got in touch with the Department of Natural Resources, and I said, hey, I bet you have a database of wastewater treatment plants. Can I have it? And they said, sure. And they told me where every wastewater treatment plant is. a little intimidating and not particularly useless and not something I was going to study. In part because a lot of these wastewater treatment plants are associated with very small towns. Almost every small town in this part of the country has a small city wastewater treatment plant, most of which is being evaporated. So they take the water, they spread it out, they let the water evaporate, then they take all the crud that's left behind and use it on farm fields, they take it to the dump, what have you. So if we just narrow it down to the wastewater treatment plants that we're feeding into the Missouri and Mississippi River, what we see is there's still a lot of wastewater being fed into the Missouri and Mississippi River. And what this is also showing is it's color coding the amount of water that's being discharged. So the darker the image, that means more water is being discharged. You can see some kind of natural collections, right? Around St. Louis, unsurprisingly, there's a lot of people, so there's a lot of water being discharged. Around Kansas City, similarly, there's a lot of water being discharged. But where I was, right, in Columbia, is one of the relatively more common places to see water moving in because it was a moderately sized city. It was about 2,500 people, including the kind of metro area that fed that wastewater treatment plant. Okay, so a perfectly reasonable question is, okay, there might be cocaine, is it a problem? Right? Practically, we might want to know if it's a problem. And what we are finding, just like when we started looking for cocaine in the waterways, we have started to find cocaine in animals. So there was a study that was done in the several estuaries in London, and it turns out they were starting to find measurable quantities of cocaine even in very small invertebrates. So it is starting to be taken up by the fauna in these places. You can also do studies where biologists who apparently love to torture animals in all sorts of different ways, provided some of you are acknowledging that, started having, in this case, zebrafish exposed to constant levels of cocaine to see where it starts to accumulate, and if it does start to accumulate. And what you can see here is this is a zebrafish up at the top. Here is some imaging that is done by profiling the different chemical composition inside those fish. And part of what we see is, oops, wrong way, right? Part of what we see is we start to concentrate high levels of cocaine in the skulls of zebrafish. It is accumulating in the tissues. And once we start accumulating things like cocaine in the tissues, a reasonable question is, does it matter, right? Okay, it's there. We found it where we've looked, whether that's in water, whether that's in fauna, does it matter? And what we can do is we can then slightly change how we torture our animals, right? And look at locomotor activity. So this is how active are these fish when exposed to cocaine. And part of what I'd like to point out is up top here, we have a control group versus a relatively low dose of cocaine. And the white dots are the control group. The diamonds are the triangle. And you see at low concentrations, not a lot happens, right? When they turn the lights on and off, the fish move or don't kind of in concert with each other. But I'd like to draw your attention to the bottom right, where all of a sudden what we see is as our concentration gets higher, right? So this is 5.6 micromolar. That'd be a really high concentration, but you know, that's neither here nor there. We start to see these significant divergences in how active the fish are, right? As a result of being drugged. We have effectively drugged our fish, and it turns out their behavior has changed. Now that might not matter to me in the zebrafish, right? But if we start thinking about what other potential types of damage we could do, we could start to be concerned potentially about what's going on. So if we look at zebra mussels are ubiquitous, right? They make for an excellent toxicology study for a couple of reasons. One is they're a relatively ubiquitous surrogate. They show up everywhere. They are alarmingly resilient to being spread all over. The second part, no one likes them because of that invasiveness. So why not poison them, right? If we're going to kill something, let's kill something we don't like, right? And part of what there's two studies I'm going to talk about very briefly have done here is they've exposed zebra mussels to different concentrations of cocaine and studied what happens. Specifically, they're looking at damage to their DNA. So what these figures are showing is damage done to DNA. And what you hopefully notice, right, is as these are exposed for longer periods of time, right? So this is for 96 hours, you start to get significantly different amounts of DNA damage inside of zebrafish. Okay, when you do longer term studies, so now instead of measuring on the order of a few days, we're looking at a week or more, right? That exposure to benzoeconine, if you don't know what that is, worry not, we'll talk about it in just a second, right? Gets worse. So as we're exposed more, we see in fact more damage to our DNA. All right. That was the background. Everyone was still with me? Fantastic, right? So we have this problem where we do see a direct effect from cocaine and some of its metabolites. Part of how we can think about what's going on inside of these is to think about the processes of metabolism, right? And I am first and foremost a poor chemist, right? And I'm an even poor biologist, right? And so I'm going to give you the ultimate cartoon version. I'm going to very generally break things up into two groups. First are metabolites. So if we're thinking about cocaine, cocaine is ingested recreationally, right? And then it is excreted in a variety of forms, some of which are called metabolites, right? We can also think of what are called transformation products. If a metabolite is something that is emitted from an organism, say a person, right, through its urine, principally in the case of cocaine, these transformation products are other things that can happen to those molecules once they encounter kind of a broader, more diverse set of organisms, right? So if we think about human metabolism, that's what I've just described with what's happening inside of our body before it's excreted. We could also think about a more complex, because microbes, it turns out, are terribly complex, right? But there's a broader set of possible pathways for how these molecules can get broken down. And then life gets even more complicated because we can have biological transformations into different products than what we might have expected, or non-biological transformations. These would just be the process driven by environmental factors, right? If you put this into water, it can undergo hydrolysis in some cases. If you expose it to light, that light can actually provide enough energy to start breaking bonds. And part of what this leads us to is a challenge anytime we're looking for these types of compounds in the environment. Because if we think about our cocaine, right? When cocaine is excreted by people, about half is emitted as a compound called benzoeconine, right? This is the principal metabolite for human use of cocaine. And it's a relatively simple change, right? If we look at these two structures, we've lopped off a carbon group, right? Replaced it with a hydrogen. We can also have between 30 and 45% lopping off of this other ring structure, right? So we can cleave across that ring. You'll notice so far that there have been abouts. And that's not just because biology is messy, and I'll fight anyone who says there, right? But also because there's another possible pathway. And that is when cocaine is consumed with alcohol, like it often is recreationally, it can form another compound. And the secondary, this metabolite is called cocaethylene, right? It's the result of the addition of that ethanol onto our benzoeconine. And it is part of what causes the euphoria associated with consuming cocaine and alcohol simultaneously. You get the secondary effect from that metabolite. There's also some amount that just goes somewhere, right? These are minor metabolites, right? And it really depends on what's taking place inside of our organism. So with that background, we're going to start trying to understand how these metabolites are moving through a wastewater fed system, right? So my study site was done at the Eagle Bluffs Conservation area. So this is a map of the conservation area. And of important note is up here at the top, this purple area, that is a set of artificial wetlands operated by the City of Columbia as part of their pollution mitigation strategy. So all of the wastewater we're going to talk about moving into this conservation area has actually already been through a several day process of treatment inside of an artificial wetlands. Oh, so excuse me. From here, right, water is piped along this curved road into a distribution channel, right? And the study we're talking about today was done in the spring and summer. And so what we see is there are parts of this conservation area that were dry. It is allowed to dry out and flood naturally, and I emphasize the heavy air quotes there, because it's controlled by the conservation area as part of their weed mitigation strategy. So what we're doing is we're selectively flooding, no, I'm not. What they are doing is selectively flooding different portions of this conservation area. And I'm going and looking at what I can see. So I've sampled in a number of different places, right? The first is right at the mouth of that distribution channel. That is where the rawest wastewater is coming out. Okay. I also selected a pool that had been held constant for several weeks, right? So this is an area that had been flooded with wastewater and then allowed to sit. Okay. There are also a number of other places throughout here. So it's actually a very pretty conservation area, as long as you don't think about where the water's coming from, right? But what we have here is we have a lot of biological activity. So I sampled further down in our distribution channel. I sampled at one of the drying edges of one of these pools that was in the process of being flooded. Okay, I sampled throughout the rest of this conservation area, including at the exit where the water is then allowed to discharge into the Missouri River. Okay. Now, I promised or threatened, depending on your point of view, that this was going to be about how we can use analytical chemistry in order to make some measurements about an environmental system. So in order to actually go from having these samples, these raw water samples, into something we can test, there's a bit of a process. Okay, so we collected a series of grab samples, right? So these are samples from the surface waters, and what you can hopefully see in that photo is they contain a rich diversity of stuff in them. We'll talk about that stuff in a minute. Okay. However, that stuff would be problematic, right? So our first step is we filter them, right, to try and remove all the suspended particulate matter. We spike them with an internal standard. That's going to give us some control over understanding what's going on inside of our system. Okay. And then we undergo a process called solid phase extraction. This, as demonstrated, is not the most efficient way to do it, but it sure is the prettiest, right? So what we're doing is we're taking liters of water and depositing all of the small organic molecules down into a very small amount of material. It's called a sorbent, and we choose it so that it collects these cocaine and its metabolites. Okay. Once we have those metabolites, we extract them, we concentrate them, and we have to go through a derivatization step. And what we're talking about in terms of derivatization is, as we look at the structure of our three principal metabolites here, we see on cocaine and cocaethylene, we have these methyl and ethyl groups respectively, but in benzoecgonine, we have this carboxylic acid. And one of the challenges we ran into is that benzoecgonine represents half of the metabolite we expect to see. But that compound does not travel through GC well. So in order to improve our ability to detect that compound right there, we undergo a process called derivatization, where we convert it to this trimethylsilane product, and then all of a sudden it will much more easily move through our GCMS. Okay, so what's GCMS? Gas chromatography mass spectroscopy. It's this thing, right? It's a big box, or at least that's what it looks like. On the left-hand side, we have a gas chromatograph. And what that's going to enable us to do is it's going to separate our molecules for us. The second half, the mass spectrometer, is going to take those molecules we've separated, smash them to bits, and then we're going to be able to count how many ions out of those bits we're getting to produce something called chromatography. So this is the type of data that we would be looking at. This is for a cocaine standard and a cocaine metabolite standard, right? So our first is that small molecule, that ecgonine methyl ester, okay? We then have cocaine, cocaethylene, and finally, benzoecgonine, right? And part of what I hope you notice here is we're actually able to achieve a separation in those compounds. All of a sudden, we're able to separate how much benzoecgonine do we have from how much cocaine, from how much cocaethylene. Okay. Once we've done that separation, right, we can do some field samples. And this is what a field sample looks at. First thing you'll notice, we have none of that benzoecgonine. Makes sense. It's a small molecule. It's tasty food for all sorts of microbes in there. When we zoom in on this region where we have our cocaethylene, our cocaine, and our benzoecgonine, what we see is we're getting good resolution of those peaks, and we're able to detect them even in those environmental samples. So if we turn towards trying to understand what's going on inside the conservation area, right? Couple of things pop out. First, out of the 8 sites that were a part of this study, we only detected cocaine at 5. Okay, there were 3 sites where we didn't detect any cocaine or its metabolites. One of them was the pool that had been sitting for a really long time. So the water in that pool had been stagnant for several weeks. So it's possible some of those biological transformations or some of those abiotic transformations we were talking about are taking place and we're moving it. Okay. The other thing that you'll notice is where we were flooding our pool, right down here, we hadn't yet gotten measurable amounts of cocaine. We also didn't detect any cocaine at this exit, and it may have had to do with how water was moving within this site. The final thing I want to point out is that our other 5 sites where we did detect cocaine, we didn't just detect cocaine. In some places we got cocaine, we got cocaethylene, and then we got that benzoecgonine. Okay, keep in mind that the relative heights of the peaks inside of one of these compounds gives us some information about the concentration. Taller peaks, more of those molecules. Comparing between peaks becomes complicated because of how our instrument works. So instead of talking about these peaks and looking at squiggles for long periods of time, let's put some numbers together, right? So what we do when we account for the response of our instrument to our different compounds is as our water flows from the outlet of our wastewater treatment plant through the conservation area, we get variable levels of cocaine. Sometimes we see a lot, sometimes we don't see it very much. And this actually initially was a bit of a puzzler, right? As we're moving through our wastewater treatment plant, or excuse me, away from our wastewater treatment plant, why do we see less cocaine? Well, it turns out cocaine use is not constant. Okay, so part of what we may be seeing is an effect from variable inputs, right? How much cocaine is coming in to the wastewater treatment plant, moving through the whole treatment process, through those artificial wetlands and into our system, right? And this is something we were never able to tease out. Is this an effect from a temporal variation? The fact that we're looking at water at different points. So if we measure all in the same day, the water that's at the end has been in the conservation area for 12 or 15 days. If we're looking at the beginning, it's fresh. Okay, so this is something we were never able to work out, right? But what we'd like to know, right, is does this matter? We talked about those 4 fish and mussels, and when we think about the fish, when we think about the mussels and where we start seeing toxicity, in that chronic toxicity, we are getting concentrations that are greater than that chronic exposure. Right? The concentration of cocaine moving through this conservation area was higher than what would lead to some of those chronic DNA damaging effects that we talked about. Okay. Now you'll notice there's a couple of asterisks up there. And those asterisks are one of the problems. Okay. The challenge here is when we're working with this wastewater matrix, we didn't get quantitative derivatization. We didn't get all of the molecules that we put in consistently being converted over from benzoecgonine into our trimethylsilane derivative. Okay, part of the explanation for this is wastewater, and once it gets into our conservation area, the wetlands is full of other stuff. Right, we saw those differences in the color from our samples. And one of the possible culprits of this is humic substances, right? This is part of the breakdown of primarily plant material. And what we see is we have a whole bunch of different functional groups that can also react with that derivatizing agent. Okay. When we did a study looking at our recoveries, what we saw was when we were working in deionized water, we got great effective recoveries. Okay, we used a simulated rainwater, so we put some salts into it just to see if that would affect our products, and we get slightly lower recoveries for our cocaine and our benzoecgonine. Okay, but by the time we get a simulated river water, so now we're adding humic substances to that, our recovery drops dramatically. So this was one of the questions that I had: is what is causing this? Okay. Also, we have some complications from our samples. If we think about kind of a simplified view of what's taking place in our samples, we can imagine having the sediment, the bottom of our distribution channel, the bottom of our different fields. We have our water column, and in there we have all these suspended particles. And one of the questions is, are these metabolites, our cocaine, our cocaethylene, being moved onto those surfaces through an absorption process? And this was the next part of the project. And then two things happened. First was COVID, shut down the lab. The second was I got hired here. And one of the challenges with environmental chemistry is you need an environment in which to do the chemistry, right? And so I had to shift my focus just a little bit away from being able to answer the question of what the heck is going on with these compounds, okay? So a perfectly reasonable person might be saying, okay, Babeco, you were doing cocaine, no, you were studying cocaine in a conservation area in Missouri. How does that connect to an understanding of what's going on with the Alameda fire? I want to point out, I'm going to be talking about the Alameda fire. This is a sensitive issue. There are no pictures of the aftermath of the fire, but there are some pictures of the Greenway. So if that's going to be upsetting to you, hopefully that is fair warning. I won't take it personally if you leave. Okay, so when we're thinking about our Greenway, right, before our Alameda fire, we could identify a couple of different parts of this Greenway. First is Bear Creek. And inside Bear Creek, we have the watershed, right, from Emigrate Lake all the way down to the Rogue River. We also have on either side of Bear Creek, this buffer region of the Greenway. Right? What complicates our analysis here is, in addition to being flanked by a Greenway, we are also flanked by two major roads. Interstate 5, right, and Highway 99 run parallel to almost everything inside that green belt. Okay. And then we also have the bike walking path. Okay? So when we're thinking about what happens and how we can experience the effects of this fire, right? As the fire goes through, right? One of the questions is what's happening inside that buffer region, right? That buffer region, the Greenway, the kind of brownish boxes that labeled here, can act as a protectant for the Bear Creek watershed, right? It can trap pollutants that are moving through a combination of different species, right? So if we're thinking about, some of the soil organic matter, right? Hopefully what you notice is this looks an awful lot like those humic substances that we saw in the last slide, because it's basically the same stuff. It is biological material that is undergoing a variety of degradation pathways, right? Breaking down and providing us a whole bunch of different groups that we can actually have interact with molecules and with those inorganic contaminants to act to contain them. When fire goes through, things change, right? So the first are kind of the dramatic obvious effects. You see that dramatic change in the appearance of the Greenway. There are still burned trees standing in that Greenway, but there's also a more subtle shift. Inside that soil organic material, as the fire goes through, you can actually have a conversion of that organic material into what's called pyrogenic carbon. And one of the things you notice is our chemical structure has changed. By the way, neither of these are really what these molecules are. They're just to represent what's going on. So don't quote me on that particular molecule, but biology is messy. I think I've said that 4 times now. I'm aiming for 6. Okay, so as we look at that molecule, part of what has changed is we've lost some of those functional groups that can grab on to those contaminants. So immediately after the fire, there's a concern that there's going to be more movement of contaminants from both 99 and I-5 into the waterway. Additionally, right, there are the effects of the destroyed homes and businesses, where there is a change in material properties, both from the loss of those structures, but also from the soil around it. That happened in September 2020. I got here in September 2021. It's did like that much, right? And so the question is, when we are looking 20 months later, what do we see? Okay, how has this system changed? And I'll be completely honest with you in 2 ways right now. First, that we are still working on answering some of these questions. So what you'll be seeing today is preliminary data. We don't have all of the answers, and we're still working through some of our samples. The other thing is, hopefully there's starting to be some regeneration and rejuvenation inside that greenbelt. But what we're going to try and address today are a couple of questions. First, is there evidence of increased level of pollutants inside the Greenway? Second, can we identify sources for those pollutants? It's a process called source apportionment, and it can be a real pain, especially in a complex area. And then the third is, how are these potential pollutants impacting our ecosystem? Okay, and we're going to look at using a relatively straightforward microbial analysis to get a general sense of what's going on inside of our soil. Now, to do this, I was very fortunate to have 3 fantastic students last summer work with me. And so what they did is, first they created my favorite like bad band cover photo underneath I-5 at one point, but they went through the Greenway and collected a total of 65 samples. These were collected immediately adjacent to the creek, and then depending on the location, 60 to 100 feet away, to get a sense of what's going on inside the Greenbelt. This involved a week of them walking from Ashland to Medford, stomping through the brush. And I'm not gonna lie, one of the best parts of this new position I find myself in is the first day I taught them how to sample. It was great. Second day I watched them as they sampled. Third day I dropped them off to, I'll meet you down there when you're done. And you know, they were very helpful and agreeable in that process, right? So we collect, well, no, let's not get ourselves. They collected all these samples, in addition to collecting through the entire burned region, really from right next to the wastewater treatment plant down here in South Ashland, all the way through into Medford. We also collected a number of background sites, just to get a sense of what's going on in our local environment. So we have a handful of sites we're calling rural background sites. These are areas principally away from any kind of industry. It is away from homes, and it is away from recent fire activity. We also collected a number of sites from Interstate 5 to get a sense of what does pollution along Interstate 5 look like. Okay? Finally, we did go into a couple of places where there was still homes that had been burned, and collected soil samples in those areas to get a sense of what's going on with the structures that had burned in those different areas. Okay, once we get back to the lab, then the real fun begins, right? All our samples were stored on ice to try and minimize the change over time, right? They all had to get sifted. This is to help us remove kind of the cold material, right? They then got subsampled. And then eventually a portion of them got dried, so we get a sense of how much moisture is there in the soil. Okay. From there, we're going to look at 3 different pieces of this project in order to address these 3 questions I've already talked about, right? We want to know what's there, and we want to know if we can identify those sources. To do that, we're going to look at a variety of heavy metal contaminants and a group of molecules called polycyclic aromatic hydrocarbons. We're also going to try and understand if we can get a sense of what's going on in the ecosystem through a microbial activity test. Okay, so for these heavy metals, right, this work was done by Gina, and it involves taking another sample, doing a digestion, doing so much filtering, so much dilution, and eventually inductively coupled plasma optical emission spectroscopy. An analytical chemist, I'm going to talk about the analytical chemistry for just a second. Here, what we're doing is we're putting our liquid extracts through a plasma, right? That plasma provides a lot of energy. It excites our atoms. As they move out of the plasma, they have to get rid of that energy, and they release it in the form of light. And we can measure that light. And by measuring how much light we're emitting, we can actually get concentration data. Now, in terms of where we are sampling on our periodic table, we were looking at some of our transition metals, right? So we were looking at, and one alkali earth metal, whatever, right? For primarily in this transition metal region, right? And one of the things that we saw that, I'll be honest, made me a little comforted, is we could not detect beryllium, arsenic, selenium, or cadmium. The levels were below what we can detect with our instrumentation, and that's great. Some of these are real nasty, right? So not being able to see these, fantastic. But I mentioned this idea of source apportionment, trying to identify where different contaminants are coming from. And we can do that by looking at how different metals, in this case, are moving and how they're moving together. So do the concentrations of these compounds go up at similar rates? Do they go down at similar rates? And some of these are associated with asphalt wear. So if we think about that green belt, because it's bordered both by Interstate 5 and the Greenway, which is primarily an asphalt way, one of our questions is this the result of weathering from that asphalt, whether it's I-5 or from the Greenway? Right. However, some elements like copper, right, are problematic because they can have more than one source, right? So when we're thinking about source apportionment, we can think about copper being associated with brake dust, but we wouldn't just want to look for copper. We'd look to see, is it moving with manganese? Is it changing with some other elements to give us a sense of if that is associated with brake dust, or is that associated with tire debris, or is that associated with our asphalt, or is that associated with okay, because there are these multivariable ways that our pollutants can enter our system. Now, I said at the outset, one of my goals was to understand what is happening in terms of pollution in this environment, right? So we can calculate something called a pollution index, okay? It's a simple ratio of how much of a particular material there is in each sample site and compare that to our reference sites. And what we see is when we look across here, right, so these are all of the metals where we actually detected something, right? If we're kind of at this low between 1 and 0, this is a low pollution relative to our background. If it's less than 1, that means it is less polluted than our reference sites. And what we hopefully see is there are some places where we have lower levels of pollution than our reference sites, okay? We also see that we can have what's considered moderate pollution, right? In a pollution index that's between 1 and 3, right? And this is where we might be concerned. We can also think about high pollution levels relative to those backgrounds. So we're going to real quick take a look at cobalt, right? Because when we look at cobalt, part of what we see is we have this reduced amount for our Creekside samples and our Greenway samples. We're going to look at chromium, copper, and then lead, because lead's just weird. Right, lead's doing some goofy things. So when we look at cobalt, okay, these are box whisker plots. These are showing all of our samples. I apologize that they're a little hard to see, especially for our structure fire background. But hopefully part of what you can see is there's a really wide distribution of concentrations for cobalt in these systems. And part of what that tells us, right, is that's not unsurprising, right? We're working in a natural environment. And one of the problems with working in a natural environment is there's high variability. So that's part of why we turn to things like our pollution index to get a sense of how things are changing. What we see for our I-5 background right here, there's about there, right? We see relatively high concentrations. We have a really wide distribution of concentrations in our rural background, right? For our rural fires, so these are places that experience the Alameda fire, but we're not near homes, right? To get a sense of what's going on in those ecosystems. And then for our structural fire, we see both a higher mean concentration and also a continued wide distribution. Once we have these data, we can then start to see, are there differences? Are there meaningful differences, statistical differences between our different samples? And that's what this bar up at the top is talking about. And there is a significantly lower, like a statistically significantly lower concentration of cobalt Creekside versus our Greenway. Cool. There's less pollutants closer to Bear Creek. I'm thrilled by that. When we then look at chromium, right, we see some similar trends. Here we have much higher concentration of our, in our structured fire, but because of the relatively small number of samples we have, we can't say that they're necessarily significantly different, right? It's a problem with not having even having a total of 65 samples, we didn't have enough data, and that's just science for it. It's a real adventure, okay? So we have these higher levels in our structured fire, but we're not able to say that they're significantly higher. When we look again at our Creekside versus inside of our Greenway, what we notice, right, is again, we have significantly lower concentrations immediately adjacent to the creek. When we get to copper, things change, right? So in the case of copper, we have higher concentrations in those structured fire areas, which might make sense if we think about what those primarily homes are composed of. Some of them have copper pipes, some have copper wire. There is more copper present that might then be distributed through those samples. And that's what we see. We see these elevated levels of copper inside of those structure fires. But because of the wide distribution and the small number of samples, we can't distinguish between our other sample sites. Now, this one point that I apologize, these dots show up so well on my screen in my office, but now you can barely see them. There is one very high level of copper. It is in fact our highest concentration of copper, and that is at the Phoenix exit, where the West Valley View Road crosses Bear Creek. This is immediately up from that, inside that park, whose name just out the back of my head, right? I can't explain why, right? And that's going to be, I expect, a perhaps disappointing answer for some of you, that I won't be able to tell you why. What was special about that point? Did we just happen to hit copper that day? And we are going to be puzzled over that one data point for a very long time. Okay, this is the power of statistics, right? We have that one that's our highest concentration, but because of where the rest of our data fall, it's not going to throw off all of our data points, and statistics speak it's an outlier. But that's a topic for another day. Please don't take analytical chemistry next fall. Okay, so if we're going to look at lead, we have a very high distribution of lead, principally in our rural background. And again, we have this kind of weird point, right? Our highest measured concentration of lead was in our rural background. Where it was located is up Highway 66, right? And there's, in my mind, two possible explanations for this. First is an ongoing historic contribution from leaded gasoline. When studies have been done in areas where leaded gasoline has been used, you consistently see higher levels of lead, and it takes a really long time to remediate that, okay? The other one, given, again, this particular site's location, is it's on the approach to Ashland Airport, coming from the south. And a recreational aviation gas still contains lead, right? So one of the questions is this a result of being in that flight line, or is it just weird and we got a weird sample, right? And unfortunately, the way to find out is to go take a lot more samples, and that's a project for another day. Okay, but hopefully what you're noticing is we have a ton of data, right? We've seen a lot of data points, and there are at times somewhat conflicting information. So we can again turn to our friend statistics, or our kind of slightly obnoxious foe, right? And we can do a technique called principal component analysis. Principal component analysis is a data reduction technique, where instead of looking at all of the data, right, we had 65 sample sites. We analyzed 9 metals, right? That is a lot of data to try and put in a nice little bar graph, right? What principal component analysis does is it allows us to reduce our data into some loadings, and that's going to say how our different components contribute to our uncertainty that we observe. And part of what we see here is when we get these arrows are corresponding with different elements that we measured, right? These are all of our data points. And if we look at our horizontal axis, it's called principal component 1. It accounts for about 56% of our variation, right? Our principal component 2 accounts for about, if I'm being generous, 15%, right? And so between these 2 points, we're explaining about 70%. No, we're explaining, yeah, about 70% of our variation, right? Which is not great. I'd love it if it was like 98% of our variations. That's very clean data. That is not environmental data, so I'm thrilled. I have a kind of good understanding of where these are coming out together. Part of what we see is if we think about what we'd expect for the 3 types of sources we talked about. If we think about asphalt wear, brake dust, and tire debris, we would expect our lead, our nickel, and our copper, and our chromium, right, to all be moving together. They'd be going up together and down together based on their source, but that's not what we see, right? So one of the implications of this is these 3 possible sources that are really strongly associated with the interstate and Highway 99 are probably not our principal sources, okay? Because what we'd expect to see is these 2, our lead and our zinc, moving in combination with some of our other elements, and we don't see that, right? So what does that mean? Well, when we take kind of the totality of all this evidence, right, there are a couple of things we can kind of conclude. First, there is a huge range of concentrations of these metals. That's not surprising, we're working in the environment, okay? The second piece is when we're along Bear Creek and when we're in the Greenway, those levels are typically lower than both the fire impacted region and I-5, and I'm going to call that a win, right? We are not hugely contaminating that green space with these metals. Okay. The other factor that this points at is there's not one source for this contamination. There's lots of possible pieces that are feeding in. It could be some of the fire activity, it could be some vehicular activity, but there's not one single thing that we can say, this is what's wrong, let's go fix that. It'd be really nice if it was, but that's not the real world. So if we're thinking about our kind of 3 questions, what is there? How can we identify sources? We've done that with our metals. We can then think about our polycyclic aromatic hydrocarbons. That's a mouthful, so I'm not going to say it too many more times because we abbreviate these as PAHs. And what PAHs are, as the name implies, multiple rings fused together, right? So if we have 2 carbon rings fused together with hydrogens, this is naphthalene, right? If we have 3 rings, we get a whole bunch of different conformations, 4 rings, et cetera, et cetera. Where do these come from? Well, overwhelmingly at this point, the most common source for these is incomplete combustion, right? So as you're watching that black smoke being emitted for someone rolling coal, part of what you are seeing in that is that incomplete combustion, and that's going to contain a huge number of these PAHs. Okay, I'm going to take that away because it bothers me. Okay, part of why we care about these is they also occur naturally in fossil fuels. So we can imagine having some of these compounds being emitted into the environment from gas leaks, from diesel leaks, in a location with heavy coal deposits. You can measure the effect from those coals. Why do we care? Well, it turns out these are toxic, right? They are both acutely toxic, especially when we're thinking about the smaller rings. These are able to penetrate deep into the lungs through small particulate matter. They're taken up and can be acutely toxic. Our larger, our high molecular weight rings are also carcinogens, with seven of these being identified as EPA priority carcinogens. Okay? So we have this question of, are these compounds present in Bear Creek because they can give us information about the fire? And it also turns out they're nasty and you don't want them. Okay, so if we do a similar technique, right? So again, we're going through an extraction process. We were very fortunate to have donated last year funds to purchase a microwave digester, which took what would have been a day-long process per sample and sped it up to about 30 minutes to do 12. And it's fantastic. It's the only way I was able to do this project, right? You then, just like before, filter them. And in this case, we actually condense it. We want to get that large volume we're extracting in down to a small concentration so that we can see what's there. Okay? Again, we use our friend the gas chromatograph. So here we have separation of all of our different compounds, and we can take advantage of a mass spectrometer, the thing that's looking at those different ions that get produced to identify all of these different components. So if we turn to our friend the pollution index, we're going to look at our low molecular weight and our high molecular weight compounds. And part of what we see in our comparison, right, is in the creek side, right? So that's all the way on the left of that first panel, right? And in our greenway, we have higher concentrations, certainly higher concentrations than our rural background. We also have very wide distributions of concentrations for our high molecular weight. Remember, those high molecular weights are the carcinogen that we'd be concerned about inside of our environment. Okay. Now, pollution index. I moved slides and I forgot. I apologize. This one's the one that scared me. Okay, because before we talked about the kind of low, medium, and high, and if you're having a hard time telling what's what, let me help you. Right, our low molecular weights, that's on my side, high molecular weights on the upper side, right? And if we come on, there we go. If we go ahead and draw in that 1 and 3 line, right? That one line, if we're below that bottom dotted line, that's good, right? If we're between the green line and the orange line, that's where we're thinking about moderate levels of pollution. Part of what we see is we have one compound, benzoanthracene, where we have very high concentrations of this PAH. Okay? And when we dig in and actually look at those concentrations, overwhelmingly where we see the highest concentrations are in our I-5 background, right? Those are those sample sites left right along I-5 where we're getting large concentration of this carcinogenic PAH. Okay? We also see, however, that we do have elevated levels of these PAHs compared to our rural background, but it's only significantly different than our I-5 background. I guess that's good, right? I guess it could be worse. Yeah. Structure fire, still a problem, right? Rural fire, you can't even see the dots because they basically overlap, right? And this work we're still doing, so we're missing a few data points compared to what we had for our metals. Okay, so we have again another kind of challenging problem in that we have this high level of a particular carcinogen, right? But we can also use our pHs for what's called source apportioning, right? Identifying the sources, because we have two different types of compounds. We have thermodynamically stable compounds. These are going to be the ones that when formed stick around for a really long time based on thermodynamics. We also have kinetically formed ones. These are formed fast in high temperature systems and over time will either degrade or be converted. So we can actually choose pairs of these compounds that have similar molar masses and figure out what's going on inside of our environment. So here we have anthracene and anthracene plus phenanthrene. And when we look at the ratio of anthracene to phenanthrene, we can start to say, hey, if we have a lot of our anthracene, that's indicative of having large amounts of combustion. If we don't, that might mean that we have a lot of fossil fuel contamination. Okay, we can do the same thing with other pairs of compounds. So what we're about to see is we're about to see a whole bunch of plots talking about different pairs of ratios to help us with our source apportionment. Okay, so this is our first one. Okay, I'm going to go through this because we're going to see the horizontal axis isn't going to change, right? So when we're on the left side of this axis, that's indicating that we have high contributions from fossil fuels. When we're in the middle, that's vehicular emissions. And when we're on the right side of these dotted lines, that's associated with grass, wood, and coal combustion. Okay? By itself, this one type of data doesn't tell us everything. And that's why we can use these pairs of compounds, because when we're looking at anthracene versus phenanthrene, right, what we see is when we're above this dotted line at about 0.1, that's typically associated with combustion of all types. When we're below that line, we're starting to think about the effects of our fossil fuels. So what do you notice in here? What do you notice? This is the audience participation point, because I noticed some of you nodding off, and I'm not, the teacher in me can't help it. What do you notice?
The Greenway data is limited to the vehicular.
All right, when we look in this part in the middle, right, we see the Greenway. That is our kind of orangish dot. We have this reddish dot. Both of these are really strongly associated with vehicular emissions, right? Our pollutants are coming from vehicles. Okay. Also comforting to me, we don't have a huge contribution from fossil fuels. If we did this after an oil spill, we would see all of our samples down in that bottom part. By itself, one pair of these ratios is an indicative of what we can expect. So we can look at other pairs, right? So here we're looking at our benzoanthracene and chrysene, right? Again, we can see on the top a strong influence from combustion, right? Down toward the bottom, we have in our middle, we have our mixed sources. All the way down here at the bottom, we have our fossil fuels. Okay, this is kind of in contrast to what we saw last time, right? In our previous set of molecules, we saw that really large concentration, excuse me, of points above, not in the fossil fuel region. My best explanation for this is we're just getting overwhelmed by that really high concentration, that one pollutant we saw with that huge pollution index, that benzoanthracene. Okay. Finally, when we look at our larger molecules, right, again, we're looking at different types of combustion sources, and overwhelmingly what we're seeing is petroleum combustion contributing the largest amount to our observed levels of these pollutants. Okay. Finally, we can do the same data analysis, or excuse me, data reduction technique, principal component analysis. And part of what you see, right, is all of our small molecules, right, our low molecular weights are all kind of going in one direction. That could indicate they're all from one possible source. There's another possible explanation I'll talk about in 1 second. Okay. We also see all of our carcinogens moving again in the same direction. Okay. Here we're explaining about 70% of our uncertainty again. And as an environmental chemist, I will take that any day of the week. Okay. That's doing a pretty good job explaining where these different components are coming from. Okay, what does all this mean, right? So just like before, we can draw some conclusions. First, right, we see elevated levels of those carcinogenic pHs inside of the Greenway and along I-5, specifically with benzoanthracene having very high concentrations, especially along I-5. Someone was asking me, what's the point of your talk? I told them, don't eat the dirt, Okay, don't eat the dirt along the highway. Don't eat the dirt in the greenway. That's not gonna serve you well, right? We also see that there is some strong evidence for an influence from vehicular exhaust, right? And finally, there are probably different sources that are contributing to these different molecular weight, our low molecular weight and our high molecular weight. Now, I've ostracized that and I said there might be another explanation. That other explanation gets us back to that idea of transformation inside of our environment. And it's going to bring us to thinking about our microbial activity. So when we're thinking about microbial activity, this is going to be a generic test for all the microbes, right? We're going to see the effect of fungus, we're going to see the effect of bacteria, right? And the way we're going to do that is by looking at a compound called fluorescein diacetate. When you make a solution of it, it's kind of clear. When it undergoes hydrolysis, you break 2 bonds on here to release fluorescein. Okay, what does fluorescein sound like? Fluorescent, because it has this nice highlighter glow, right? You get this very strong green compound as a result of the degradation of these compounds. So what we can do is we can actually take slurries of our soil in buffer, right? We can add fluorescein diacetate, we can incubate it. You'll notice there is a reagent link up here, right? That's giving us a sense of how much is just occurring in the several hours we're waiting for these samples to get processed, right? We can filter, centrifuge them, and then we can quantify them to detect how much of that fluorescein is being released. When we do that, what we see, right, is in the center, we have our rural background where we have our highest levels of microbial activity. Everywhere else is significantly lower than that rural background. We are seeing less biological activity in the microbial activity. I have to be careful with biologists. We're seeing less microbial activity in these spaces. And that raises a question about what's going on, okay? What my working hypothesis is that there's an effect from those PAHs. And part of the reason is some microbes can actually digest PAHs when they're provided with poor carbon sources, like you would see after a fire. So one of the questions is, are we getting different microbiomes occurring in these samples where we have high levels of polycyclic aromatic hydrocarbons? How y'all doing? Okay, good, right? So that's where we go from here, right? The obvious question is, what can we do, right? So the first thing I want to do is I want to characterize how much soil organic matter is still left behind. We're going to do this a couple of ways, okay? The first is we're going to quantify the total amount that's there, okay? We can do that a handful of ways, and if any of you are soil scientists and really want to help, come talk to me, because I don't want to do this, but I'm going to have to, okay? The second question we have is what's the difference in the composition between these different types of soils, right? And for that, we can turn to a profile done with NMR, right? Nuclear magnetic resonance spectroscopy. And what we look for, right, is we look for changes in our our NMR signal that correspond with different groups inside of there. If you look from the top to the bottom in our unburned soil, what you see, right, is you see a lot of groups associated, certainly with heavy glucose, right, and glucose, but also a lot of these are our carbohydrates, right, there are lipids, what have you. As we go into more increasingly severe burns, we're getting that conversion into these pyrogenic, these carbon-ringed compounds that look a lot like our PAHs. Okay, one of the possible problems with forming more of these compounds is this nice flat structure can act like a sandwich for our pHs, and they can actually stick through their intermolecular forces. I'm also interested in characterizing metal binding. Are we seeing lower concentration? Because all the metal just goes straight through. I don't know, but that's one of the things we're going to find out. And then I think the most important thing to do next is to actually characterize that microbiome, right? Can we see changes in the composition of our bacterial and fungal DNA that might see an expansion of the type of microbes that are acting in that soil? And some of the work that's been done demonstrates that you get a shift towards more primary nitrogen producers because all of the changes that have taken place in the soil, and you can actually see those changes by characterizing the microbiome. Okay, then what? Do it all again, right? part of what I'm interested in is how is this environment changing in the long term, right? So we've looked 2 years after our fire, what happens 5 years after the fire, what happens 10 years after the fire, to see, do we see the same trends? Do we have the same concerns? Okay. No work in science is done in a vacuum. I've been very fortunate to work with a number of fantastic people on this. So from the data that you saw today, the stuff that was done at Eagle Bluffs was collected by Vanna Couch, Stephen, Ryan, Allison, and Katie. That's Vanna recovering a sample probe that has been mucked up because working in the environment sometimes is gross. So far, I have 3 students who have contributed data to what you've seen here today. Kalia, Gina, and Carson are responsible for collecting all of the sample, and Jeff has started joining me and trying to analyze some of those PAHs, right? Again, I have to say thank you to all of my chemistry colleagues both here and at Columbia College, and I've been incredibly fortunate to be receive funding to support both our ChemRec students, that's our summer research students, through the scholarship funds. Yeah. That was a lot longer than I thought it was going to be. So, are there questions I can answer?
I know you mentioned it at the very end, but like, if you were to take more samples, like 2 or 5 years in the future, how would the data, like, how do you think data would change?
Great question. How would the data change? I don't know. Like, and that's an obnoxious answer, right? For some of those compounds, especially the ones that are demonstrated, or excuse me, that are produced from vehicular exhaust, unless there's a dramatic and rapid shift to, for example, electric vehicles in the valley, that's not going to change, right? Because we are going to continue using both diesel and gasoline engines, we're going to keep seeing those. The levels for that bisphenol, excuse me, oh boy, all the compounds that just merged in my head. That benzoate anthracene, where we have that really high pollution, that's comparable to the pollution levels you see in Turin, Italy. That's a city that's almost 1 million people, right? So it's very high. It's not as bad as I found one paper as I was looking at the data. We're not as bad as Glasgow. I guess that's something, but you know, we are seeing those high concentrations. What do I expect? I expect some of those pHs will decrease over time if they are in fact the result of fire, right? If they're the result of that vehicular activity, it's not going anywhere.
Can you remind me what your background, burn structure, samples were?
Sure, so that was soil samples collected from locations that had experienced fire that had not yet been rebuilt, right?
Not scraped clean.
I don't know, because in some places, I would expect it had been straight clean, in some places it clearly hadn't been. But I don't know, because I wasn't here to know kind of the history of some of those sites. That might explain why we see as much variation as we do in some places where we have, or we can expect there to have been some of that scraping. We'd expect it to be lower because we went through that remediation process, but I don't know the site history of all.
I'd like to talk to you afterwards, because I'm
Absolutely.
working with a group of government agencies doing water quality sampling of the same area. And we're doing dioxins and PFAS and issues.
Coming by office. Yes. For these, we only did soil samples, and we also didn't sample sediment. So sediment is inside of the creek. We were only looking at soil samples along the creek edge and further away. If we wanted to look at water, we could. Some of the techniques that we talked about with the cocaine, that solid phase extraction, we can do on water samples, get it concentrated enough, we'd actually be able to measure it, but all of these were outside. Yeah.
Is there any communication with any of the entities of the valley or governmental entities about how to be proactive with this data in terms of improving, you know, the waterways and the green areas?
I hope so. I'm just getting to a point where I have my head around like what we found, right? And that's part of why I would like to reiterate my disclaimer, especially with the PAH stuff. There's asterisks in that we are not done analyzing all of our samples, right? Some of the question around how can we be proactive? There is a lot of work in how do you mitigate the effect of storms following wildfires, right? How do you trap some of those contaminants? How do you remove them? And some of that work has already been done. And unfortunately, because of the climate we find ourselves in, these types of fires at the kind of wild urban interface are going to be more common. So it's actually something that we need to care about. Absolutely.
One more question. The microbiome is very interesting. It seems like it's changing to be nitrogen producers. That's interesting. Are they anaerobic or are they aerobic?
It depends on the soil conditions, and I'm going to defer to Dr. Stewart, who's behind you, who actually does this. I just kind of fake it, right? But what I have seen, right, is when you think about anaerobic, and if I'm wrong, just give me the sign, right? It's going to depend on the oxygen availability within the soil, and some of them it switch back and forth. But again, talk to Nick. That's why I was talking to Nick, because I'm going to get myself in a lot of trouble if I keep talking about biology. There's a lot of biologists in here.
Slowly.
Right. But yeah, that's some of what we'd be interested in seeing. What are the characteristics of those environments and how do they change in these different sample sites? Do we see a different profile in places where we have high levels of metals or high levels of these PAHs? Yeah.
So Chris, you suggested a number of times that the transport of this material is through storm events and movement across the surface. Can you talk about different mechanisms for how this stuff gets where it gets?
Yeah, absolutely. When we're thinking about PAHs, overwhelmingly the transport's particulate, right? So it's that vehicular exhaust that is being exiting out of the vehicle, right? Over time, it undergoes a settling effect and then ends up in the soil, right? When we're thinking about our metallic contaminants, that's not likely to happen, right? And so because of that, that's why we're thinking about storm events or other hydrological events, because we'd expect to see them more readily entrained in that material. But yeah, lots of ways pollutants can move. It's one of the things that makes it fun. One of the things that makes it super frustrating. Yeah.
You mentioned, I think that future research looking at soil organic matter. Are you thinking about being able to look at, okay, if there's high soil organic matter, do you have less, getting less of the pollutants getting to the waterway, or like being able to do something spatially that way?
That'd be one of the questions, right? And part of one of the questions I have is how much transformation has there been to the soil, right? How much of the conversion from those the soil organics to those pyrogenic carbons do we see, right? But along those lines, it's absolutely important to understand, does that then contribute to those higher or lower levels, right? One thing we could potentially expect to see is in places where we've had a lot of that carbon converted to pyrogenic carbon, we might see lower concentrations because it's all ended up in the creek already. Because we're sampling 20 months after the fire, right? Have we lost those pollutants and they haven't reestablished themselves, for want of a better phrase, right? Have they not migrated through our system yet? And that is one of the causes. So yeah, the spatial component is absolutely an aspect. Jim.
Yes.
Dang.
I have to go a little bit more spatial. Well, on Sometime World. So I was curious on the collection of the data. It was the summertime.
Mhm.
Were there any storm events? Was there any movement of water across the landscape during or right before?
So the previous storm event was, I think, 3 weeks before. So it hadn't happened recently, right? Well, immediately recently, right? And what we saw is relatively low levels of soil moisture in all of our locations, except a few of the creek side. And I expect at least one of them is because they fell in. But yeah, we tried to control for that, and that's part of why we did all our sampling in one kind of big nasty push. Yeah. It was also the first days of 100-degree temperature, and I'll tell you, I was really glad not to have to walk that whole way. But yeah. Other question. If you think of any, I'm going to lurk down here. And thank you all very much for coming.